So next we have Dr. Luke Jones from the University of Oxford, who's going to talk about SARS-CoV-2 neurovirulence.
Thank you. Hi everyone, welcome to my talk on the neurovirulence of SARS-CoV-2 and associated differences between the variants of concern. So, by way of introduction, at the start of the pandemic, the neurological signs of SARS-CoV-2 were particularly highly reported, things such as anosmia and brain fog. There's evidence within the literature that other coronaviruses, like HCoV-AC43 and MERS-CoV, can infect cells of the central nervous system. We now know that long COVID is a recognized syndrome, and we need to better understand the viral mechanisms that underpin it in order to have any chance with therapeutic or preventative measures, but there are lots of gaps in our understanding of this. So with that in mind, this project had a few aims. Number one, determine the cellular tropism of the SARS-CoV-2 variants of concern and see if they differ between the different variants. Quantify the replication kinetics within single cell types, and to then move on to interrogate the cellular response to infection through things such as RNA-seq or analysis of the cytokines, chemokines that are given off. So a quick introduction to the neurological side of the project as well. I am not a neuroscientist, but I'll do my best to explain this in layman's terms. We have the neurons, which obviously key, highly important. They send bioelectrical signals to each other for signaling. We have microglia, which are the resident macrophage cells of the central nervous system, pleomorphic. We have astrocytes, which perform metabolic and homeostatic tasks, things like scooping up excess neurotransmitter. And then we were very fortunate to also work with some 3D brain organoids as well, which have, as I say, a 3D structure and contain multiple different cell types and different subtypes of these cells as well, ideally. So we started off with infecting 2D neuronal cultures. These neurons are derived from human-induced pluripotent stem cells by our collaborators. 1 hour post-infection, obviously minimal viral staining, and then this slowly increases over time. So in these images, the virus is in green, and you see it spreading throughout the culture by 48 hours, but it's already present at 24. You can see it quite nicely extending down the axons. And we see this in all the variants that I've tested so far, but to different extents. We have some just higher magnification images here, just to illustrate the point a bit more. You can see in VICO1, 48 hours, it's really got around the cell body and then spread further. The green is a nucleocapsid stain, by the way, rather than spike. Also see it in Delta and in more recent variants as well, like this BA5 Omicron. So with all these images, we could do some quantification using our fancy software in the microscopy suite at Oxford. It basically looks for the green viral antigen stain and where it overlaps with the cellular stains, which are normally neuronal markers or cytoskeleton markers, and then quantifies the amount of cell stain, essentially, that is taken up by the viral stain. And with this quantification, we can see that VICO1 was the most infectious after 48 hours, but BA5 wasn't far behind. It varies quite a lot between the variants, so alpha and beta both drop down quite markedly, same for kappa as well. And then the Omicrons are a bit more variable, which is interesting. And you get sort of background level of just general crap that the stains pick up. But they're all above the background level. I also took the supernatants and titrated them. Generally, they declined to nothing over 24 hours, with a couple of exceptions. You can see VICO1 and BA5 both persisted at low levels in the supernatants until 48 hours. There's no great rise in titer. They're not being lysed and releasing loads of virus into the supernatant, but there's some interesting persistence. And it's interesting, it's the two most infectious ones as well, where we see it still in the supernatant. Yeah, all this is just residual input at 0 hours that gets washed off. I then went on to infect some 2D astrocytes, which were also susceptible to infection. And again, VICO1 and BA5 were the most infectious following the quantification. You can see 1 hour, 24 hours, and 48, it's starting to spread. These aren't the same cells, these are different wells of the plate, but the stain is increasing over time. The supernatants, a slightly different story in this one. Almost all of them persist at low levels, sort of sub 10 to the 3 FFU per mil, up to 48 hours. A couple of the strains, alpha, and I think XBB, dropped to undetectable levels. I also infected 2D microglia cultures. Microglia, as I mentioned earlier, they're a macrophage-like cell in the CNS, and these seem to be entirely refractory to infection, regardless of the variant. There was very little detectable virus anywhere in them. There was some like swallowed inoculum at the 1 hour post-infection time point, but that was it. And then that declined to nothing. And similar story of a supernatant. VICO1, it persists to 24 hours, but again, there's no great release of virus into the supernatant. So this is just a quick slide introducing the production of the 3D brain organoids, which are kindly produced by collaborators and then delivered to me to infect. The brain organoids are very expensive to produce, so we had a more limited number of strains that we used with those and fixed at 4 different time points to try and capture a more longitudinal aspect of infection. After infection, they were fixed and then embedded in wax and sectioned and then stained for immunofluorescence. And I thought this was a good opportunity to try and dig into the tropism a bit more, because there should, in theory, be different types of neurons in these 3D brain organoids. We didn't see infection in all of the organoids, particularly the cortical ones, but here you can see this is VICO1, which is the ancestral strain. Should have clarified earlier. VICO1 is the ancestral strain. And then that's got in there, essentially, and spread. So again, we looked for overlap between the green viral stain and other cellular markers, different markers on different sections. We saw the viral stain mostly correlated with mature neuronal markers like TUJ1, MAP2, synapsin, but also this PAX6, which is an early differentiation marker. Supernatant, similar story. Excuse me, similar story in the supernatant. It doesn't really increase, but it persists and slowly peters off over time. These are cortical brain organoids, and then we're also fortunate enough to work with some whole brain organoids as well, which tell a slightly different story. You can see on these whole brain ones, which in theory have a different makeup of cells, that we do get quite noticeable infection, but entirely around the periphery. Like around here, particularly in delta, it was quite noticeable. Because it's limited to the periphery and then it doesn't spread into the center of the organoid, even at 72 hours, it's still on the periphery. I'm suspicious that these are endothelial cells around the outside that are becoming infected, and that's also why we see the much better titers, like a natural increase from 0 to 24 hours. So yeah, that needs to be clarified with some sort of endothelial cell marker stain, just to confirm that's what's going on there. But the cells around the edge do look different just via the nuclei stain at high magnifications. So what are the conclusions? Neurons are definitely susceptible to SARS-CoV-2 infection by all the variants to greater or lesser extents. VICO1 is the most neuroinfectious, it seems, which is interesting because the ancestral SARS-CoV-2 was most associated with brain fog and anosmia, or brain fog and anosmia was most reported at the time the ancestral version was circulating. Despite the increase in viral staining that we see, there's very little being released into the supernatant, which suggests to me cell-to-cell spread might be occurring. Axonal transport of OC43 has been reported in the literature previously, which makes me think maybe that's happening here. There is a more recent paper that said axonal transport of SARS-CoV-2 doesn't happen, so a bit of a mixed picture. Microglia in my hands seem to be entirely refractory to SARS-CoV-2 infection, but the astrocytes are susceptible. Astrocytes and neurons are along similar differentiation paths, whereas microglia are all the way over somewhere else because they're macrophage related. So it doesn't completely surprise me that neurons and astrocytes are both susceptible and microglia aren't. Work will continue with the analysis of the multicell organoid infection and investigating the cellular response. I have some initial RNA-seq data that I didn't have time or space to include in these slides, but with that we see a much higher inflammatory response from the cells in response to VICO1 compared to the other strains that I tested. So all this sort of leads one to the conclusion that VICO1 associated high infection or inflammation levels may have been responsible for the increased incidence of the brain fog, the anosmia, the long COVID early in the pandemic, but more research needed to really drill down into that. So I'd just like to thank everyone in the Carroll group, particularly Young for his help with the RNA-seq, people at the IPSC core facility for producing the 3D organoids for me to infect, the Hand and Hetty group for growing the 2D cells for me to infect, and the microscopy core for helping train me on the microscopes, and the FDA for funding. Thank you very much. Any questions?
Thanks very much. Nice talk. Does Durase 2 staining correlate with the cell susceptibility? Do microglia not have ACE2?
That's a good question. I haven't done ACE2 staining on the microglia. I have done some preliminary ACE2 staining on the neurons, and it is expressed but at quite low levels. Like certainly in comparison to Vero's, where it's everywhere. There's low levels of ACE2 on these iPSC neurons I'm working with.
Yeah, and does this cellular infection pattern correlate with post-mortem material from people who've died with?
I haven't worked with any post-mortem material myself, but that's something I could look into in the literature.
Thanks. Really nice work, Luke.
Thank you.
Just first a comment in the pattern that you're seeing in terms of susceptibility is exactly matches what we see in the ACE2 mice.
Lovely.
But then also to say that, and you'll correct me, no doubt, if I'm wrong on this, that the cells in the brain that we see infected are neurons, and we can't detect, and you can't detect any ACE2 in those neurons. So whatever's going on isn't probably totally dependent on ACE2 in those cells, but it's interesting that pattern. And that was actually the question that I was going to ask, which is what Louise asked, is if you look for ACE2 in the neurons and levels. Yeah. But it seems like there's not a lot there. But
there's not a lot in the 2D neuronal cultures. I really need to try it in the organoids, to be honest, to see if it differs more by the different cell types that we see in there. But yeah.
It would be really good as well to see if what mechanisms and what receptors that SARS-CoV-2 is using in the neurons, because I think that would be fascinating.
There's obviously the suggestion that later Omicron variants don't use ACE2 entry as much, so perhaps that's why we see BA5, for example, taking off a little bit. But then XBB drops down again, so there's clearly more to it than just the ACE2 entry.
That is interesting, because we find BA1 doesn't go into the mouse brains at all, but BA5 does. So it matches up with it.
Hi, thank you. Very nice talk. I was just wondering, how many IPS cell lines have you used, and if these were derived from individuals or you bought them?
So I would have to ask the facilities that we worked with. I think the 2D cells and the 3D cells both came from different donors, but each of them only came from one donor. But I would have to double check. I didn't buy them myself, certainly, but they may have done to procure the initial ones that they then differentiated, yes.
Okay, because what we see in humans, that not all of them have neurological complications. So that's why I was wondering if...
Do you think that there could be donor-to-donor variation?
Could be.
Yeah.
Yeah. Okay, thank you.